biochemistry from the University of Kentucky. Dr. Haley has, was it Hal that wouldn't quit picking on you, wouldn't quit? Mm-hmm. Hal Huggins that just said, test this stuff, check it out about mercury, and maybe not. Anyway, Dr. Haley has a reputation of honesty and straightforwardness and integrity that is beyond question, and I'm thrilled that we're here to share with us today.
Thank you, Bill, and I'd like to thank the members of the IABDM for inviting me to talk. And most of you know me. I'm basically, I can't talk any louder than this. Is this thing turned up? Is there a way? Okay, they'll get it adjusted. But basically, I think it's important to follow a little bit of my history. I was funded by NIH for about 25 straight years on the same type of grant, on the same grant, same title. And you don't get that done without knowing what the hell you're doing in the field of chemical synthesis and biochemistry. It was only at the end of it when one of my graduate students took a postdoc in the Center on Aging and was working on an Alzheimer's project. And we did a very simple study, which we're going to talk about today. This is set off of Firestone. And that is that if you look at the technology I developed, it's called nucleotide photoaffinity labeling, and it allows you to pick out different enzymes and see if they change in a normal versus a disease state. And so we've done that on cancer, and it worked well. Nobody said anything. Different ATP binding proteins in cancer tissue versus normal tissue from the same source. But when we looked at Alzheimer's disease brain, we saw phenomenal differences. And they happened to be in a couple of enzymes that I knew a lot about, that I had determined the active site sequence of tubulin, creatine kinase, and glutamine synthetase. All three of those enzymes were dramatically inhibited in Alzheimer's disease brain versus normal brain. And you wonder what they would be. And being a chemist, what I knew is that each one of those enzymes had a very active sulfur in its binding site. And so I did a test, in which I'm going to explain today, but I want to give an overrun of everything that comes down to this. We published a paper showing that mercury, and only mercury, could induce the same aberrant biochemistry with regard to several key enzymes that were inhibited in Alzheimer's disease brain. And those differences could be mimicked only by mercury. You couldn't put iron, lead, cadmium, or anything else into it, especially in the presence of organic acid chelators such as citrate, glutamate, or EDTA. And so we published that. And I thought everybody would look at that and say, well, this is interesting. Mercury is a known toxin. It's a neurotoxin. Every chemist knows that. I never realized the kickback that I would get. And I want to tell you, if you offend the American Dental Association and the sources that like putting mercury into our vaccines or into our mouths, they will come after you, and they're not nice. They don't want to discuss it. They have never invited me to a meeting, which I would readily go to, even today, if they wanted to have a debate with me at the at the National Institutes of Dental Research. I would go there, and I would get on the same stage with them and fight with them. Because the science is simple, it's straightforward. You don't have to be a rocket scientist to understand it or believe it. What you have to be is you have to have a criminal mind to say that this isn't existing anymore. And so I'm not very nice to these people, and that's kind of out of character for me because I'm usually a very pleasant person. But let's just talk about what I think happens. And it's not I think. Sometimes you have to say I know happens. And I know this because I've followed the work of people like Mike Ashner and his wife, who do a lot of work on mercury vapor transport, and other people, plus common sense, common chemistry. And this is on the inhalation of mercury vapor, which comes off the dental amalgams, which people like, David Kennedy and many other people, and now the movie has come out with a big push from the IABDM to show that mercury vapor coming off of dental amalgams, especially when you're grinding an amalgam filling, mercury vapor, and we know it goes into the lungs, where 80% of it is absorbed, transported to the blood. And this mercury vapor is non-toxic, it's non-reactive, and it sits there as Hg0, and then it gets into the erythrocyte, where there's an enzyme called catalase, and other enzymes, by the way, but primarily catalase, can convert Hg0, which is non-toxic, non-reactive, to Hg2+, which is one of the most toxic metals or elements in the world. I mean, it will kill you. It will kill every enzyme it interacts with. Well, not kill every one of them. Sometimes it activates some of them, which is bad. But this Hg2+, the Hg0 can also cross the blood-brain barrier with a great deal of efficacy. If Hg2+ is produced out here, it has a very hard time getting across the blood-brain barrier and doing any problem in here. However, when it does cross the blood-brain barrier, catalase again can convert it to Hg2+. And what happens here is that Hg2+ can bind to different enzymes which have sulfur groups on them, such as in the iron sulfur centers of the electron transport system. It's also picked up by glutathione, but it can totally wipe out the amount of glutathione in your body very quickly. But what it really can also do that most people don't talk about is it can displace iron and copper from the electron transport system, from other enzymes. And once these metals are displaced, you now get what's called the production of hydroxy free radicals. These are called redox active metals. Mercury is not a redox active metal. It does not give up electrons to water to make hydroxy free radicals or superoxide anion. The chemistry just is not there. So why is it that mercury is the biggest inducer of hydroxy radical formation? That's because mercury displaces iron and copper, and when they're displaced inside the brain, they start oxidative stress that leads to many of the problems you see in Alzheimer's disease, Parkinson's, etc. It's very straightforward, and it's chemistry you can't refute. I mean, you can't say no, this can't happen. It does happen. And the most important thing here, as I'll point out, is that when iron is displaced from the iron sulfur centers of the electron transport centers, you stop making ATP. You stop making ATP, and the cell's going to die. You'll go through apoptosis, artificial induced apoptosis, we're going. And so this is all established, it's all published, and I would point out on all my slides, if you get them, I give them to anybody that wants them, there are references to tell where these figures come from and where the discussion comes from. Pardon me. Okay. This was presented, some of this early data I presented about 20 years ago to the International Academy of Oral and Medical Toxicology. They invited me to a meeting in Chicago, and I, being a chemist, biochemist, I thought they wanted to hear about how you generate free radicals or take azides and convert them into nitrines and label the active site of enzymes. I thought they were interested in the toxicology of just my chemistry. And I talked to them, and I didn't know they were dentists. I really did not know they were dentists when I walked into the meeting. And I thought I'd gone into a house of crazies. I've said that about several of the IABDM, because they were telling me that our government would put something in your mouth that would release mercury for 20 years on a regular, steady basis. And I said, no, they wouldn't do that. Our government that has a job, the FDA has a job of making sure things are safe, and you cannot put out products that release mercury. I didn't believe them. They drug me down and gave me demonstrations of people there with amalgam fillings, and it was very convincing if you believe them. And I didn't, because I'm a very suspicious person. I'll be very honest with you. Somebody gives me something that I think makes me have to disagree with the basis of what I consider honest behavior in people. I basically don't believe them. And they tested my own mouth. I had 4 amalgams at that time, and I would tell you I had a wife that had 15. And they used their mercury vapor analyzer and showed me that these people, if they chewed gum, the level went up in their mouth. And I thought, this can't be. Well, if this is true, it's going to be easy to get this job solved. You just have to tell the government they're honest. They're going to do something about it. Yeah. Well, I learned a lesson very quickly. But anyway, I didn't believe them, but when I went home, we have an OSHA department at the University of Kentucky, and I went and got their mercury vapor sniffer, and I started checking everybody in my lab. And there was one girl in there, they started calling her metal mouth. She cried. I mean, but she pegged that meter, especially if she chewed gum. I mean, she was getting a major dose of mercury every day. And my wife, like I told you, and I'm not exaggerating a damn bit, 15 amalgam fillings that were all replaced very, very quickly after I saw this data. And I had to pay for it. And I didn't mind it at all. But the bottom line is, when you see all this stuff, when you can take a mercury vapor analyzer that OSHA uses to shut down the chemistry laboratory where I was chairman, if you have it coming out of a sink or somebody breaks a thermometer, and then they're telling you that this is safe when you got it in the mouth and you can measure with a meter with chewing, and it's coming out every second of every day that you have them there, you know there's something wrong. I would tell you right now, my conclusion over this thing is that the United States government, the dental branch of the Food and Drug Administration, is controlled by criminal activity. I don't know the source of it, but you cannot deny this data. And so to sit there and say, oh no, there's a scientific debate going on. There is no damn scientific debate going on. You have to be a friggin' idiot to say that mercury doesn't come off of a dental amalgam at levels that are toxic to a human being, and that it should not be there, it shouldn't be placed there. And I would add to that, anybody that thinks it's okay to put 12.5 micrograms of ethyl mercury into a baby on the day of birth is a criminal, or an idiot to the point they should not be allowed near anybody to treat them with, for medicine. Okay. Okay. And now I just want to show you how simple and straightforward this is. This is an immune fluorescence assay showing you the amount of tubulin that's in one neuron. This is one. Pardon me. This is one neuron. I keep hitting the wrong button here. I'm sorry. This is one neuron, and this is all the tubulin that's going out. All the neural fibrils coming off from one neuron right here. And you can see it. It's loaded. It's the most abundant protein in the brain. And it is extremely sensitive to mercury. When you purify it, and you can purify it, I have purified grams of this in my laboratory because I did a lot of studies on tubulin. I mean, when I was at the University of Wyoming, way before I ever went to the University of Kentucky, and we measured the chemical activity of this. And what we can say about tubulin is lining the entire axon, and it looks like a tube. And this is microtubule here, these little spots here, and they polymerize and they form these bodies that are long, and they're actually the railroad tracks that take neurons or take bound organelles that are loaded with neurosynaptic transmitters from the synapse all the way out to the end of the axon, where it's released to activate the next neuron. It's incredibly important for neural transmission, and it's this neural transmission that is destroyed in Alzheimer's brain. I mean, your synapses disappear, your tubulin depolymerizes, and it falls apart. And this is what we're going to talk about that we saw in the brain. Now, this is a graphic, and this is a graphic to show you some of the things how this fits in with Alzheimer's disease. This is the tubulin up here, and it surrounds, and this is the one-dimensional look at this, but it surrounds all the neural fibers, the neurofibrillary that form the neurofilaments that form the neurofibrillary tangles in Alzheimer's disease brain. Formation of neurofibrillary tangles is the major diagnostic hallmark of Alzheimer's disease. To form the neurofibrillary tangles, you have to get this tubulin off of them. And the tubulin is held to the neurofibrils by a protein called tau, right here. So this connecting protein is called tau, and it is abnormally phosphorylated tau that is one of the other diagnostic hallmarks of Alzheimer's disease. In other words, it's hyperphosphorylated. And you can't hyperphosphorylate it if it's connected as it is in here. So when you dissolve all of the tubulin, you release these neurofibrils, and they form a neurofibrillary tangle, and they release this tau so that the tau does not get dephosphorylated properly, and you get hyperphosphorylation of tau. And so the only diagnostic hallmark that's not shown on here is beta amyloid protein buildup, the production of amyloid plaques. And I'm going to show you later how that works and how mercury induces the formation of amyloid plaques. It's all science, done by somebody else. But what we can look at here, and you can see this, is that the breakdown of the tubulin on the AD brain, on the synapses, would lead to the destruction, the formation of neurofibrillary tangles, and the abnormal phosphorylation of tau would be expected. And so you say, well, how did you do this? Well, this is what I'm really well known for. This is the tough part of my research. Looking at mercury toxicity is simple research. I made these compounds. They're called nucleotide photoaffinity probes. And what it is you take an analog like GTP, which is used to polymerize tubulin, and you add an azido group to it. And that azido group, if you do it properly, it will, in dark, in the absence of light, will mimic GTP. It's just a very small addition to the molecule. However, if you hit it with a photon of light, it literally explodes, generating one of the most reactive chemical intermediates known in chemistry, forming a nitrine, a nitrogen with 6 electrons in the outer shell, and it binds on whatever it's sitting next to. If it's not sitting on a protein, it reacts with water. It's that reactive. And so this was what I was funded 25 years for, was to make this type of compound for studying biochemical active sites of nucleotides such as ATP, GTP, and cyclic A binding proteins. They were invented in my laboratory and used to study tubulin way before I ever looked at AD brain. And we published in the Journal of Biological Chemistry that the beta subunit was a binding site for GTP that causes tubulin polymerization. We were able to demonstrate that you had to hydrolyze this GTP to GDP, for the polymerization to occur. And we identified the guanine-based binding domain peptides of GTP on the beta subunit with J-RIM and Haley in biochemistry. And what we found is that this is right next to a very reactive sulfur group. So sulfur is very important in the structure of tubulin. And we had published in several major journals that GTP could substitute, azido GTP could do this. And so here's the first study we did. What we would do when they did autopsy samples, the people doing the autopsy would take a section of every brain and put it in a little tube for me, for my laboratory to go pick up. And we would do it. And we would do this type of study where we would see which ones bound to GTP. You can see some of them didn't bind it at all, some were really low, and some of them bound quite nicely. So we'd screen all of these, and we'd take them back, and when the diagnosis was done, these were potential AD or demented brains. We thought most of them would be Alzheimer's. Some of them weren't. They were Pick's disease. Pick's disease does not have tubulin, doesn't form neurofibrillary tangles, and doesn't have that loss. So everything that we did here worked out well. And you can see on the normal brains, the tubulin was always very, very high and very capable of binding to GTP. Some of the demented brains had totally lost the activity. Some of them were dramatically 80% or so reduced. And so what we saw was that what you would expect, that tubulin, the axons in demented brain, were dysfunctional, and that the tubulin was not behaving as it normally should. So we checked it to see if they were there. And this is just a stain, and what we're looking at is that both the alpha and beta subunits of tubulin were found in equal amounts in normal and AD brain. In other words, they weren't disappearing, they weren't being chewed up, they weren't being destroyed, and it wasn't that you weren't making them. They were there, something had happened to them. And here's an immunostain to quantify it for the beta subunit, and what you can see is that the beta subunit doesn't make any difference if it's control or Alzheimer's. This is from that same slide. I mean, the level of the tubulin was there, so it wasn't disappearing. And so something was binding to it, making it not destroy, but not be able to function properly. And what we did is we did a simple centrifugation, and if you look at this, what this shows is that in normal brain, this is a brain homogenate. This is the staining of the beta subunit. In the homogenate, that's the brain, that's the solid part. We just grind it up. And here's the supinate, and here's the precipitate. And you can see that in the soup, of all of the control samples, there's tubulin that's soluble. In the AD, none of the tubulin, or very little of the tubulin, is soluble. In other words, it's all is aggregated, and it goes down when you just do a simple centrifugation. You know, the most simple laboratory in the United States could repeat this experiment and show that tubulin is abnormally aggregated, and that's what metals do to tubulin. They abnormally aggregate it, and it goes down when you put it in a centrifuge and you spin it fast enough. And so, and this is not something that you have to be a rocket science to understand, and it fits into what we're saying. So we looked at it, and we did a study on the effect of all of these metals, mercury, cadmium, copper, and zinc. We did everything, but that's all on this particular job. And if you take pure tubulin, all metals will inhibit its binding, all heavy metals, if you go high enough. But if you look at this, you can see that mercury does it much better than anything else. I mean, you know, by the time we get to 2.5 micromolar, we've diminished it quite a bit. Cadmium, you have to go out quite a bit higher. You have to go to 5. Copper, you have to go even higher. You have to go out to about 10. And with zinc, you have to go up to 25 to see an effect. However, you know, in the tube, with pure tubulin, this doesn't mean much. So, because in your body, you're loaded with citrate, you're loaded with fumarate and malate. These are all organic acids that have high affinity to metals. And you don't have free copper, you don't have free iron in your body. Your body works very hard at keeping iron chelated with either iron-binding proteins or with metals like this, because if iron's not chelated, it's incredibly toxic to the human body. So if we look at this here, and when we did this study, this is done to explain it to people why you would do this. This is the rate. You can see this is mercury inhibits the binding of tubulin because it has more affinity for the sulfur groups than does cadmium, than does copper, than does zinc. However, if you put in EDTA to mimic glutamate or citrate, you can use citrate. I could have replaced citrate here because it's a heavy metal chelator. You see that all the rest of them, there's a huge protection, and actually it pulls off more of the metals on there than we think, and it actually kind of increases the photolabeling or the ability to interact with it. But with mercury, when you chelate it, it even becomes more toxic. I mean, it inhibits faster, so you cannot. The take-home lesson is the organic acids that are normally in your body that protect you from heavy metal toxicity don't work with mercury. They work with cadmium, they work with iron, they work with copper, but they don't work with mercury. So here's a study on the normal brain. And what we're doing here, we're taking cadmium, mercury, cadmium, and copper and zinc in the presence of a huge excess of EDTA, which is a chelator for heavy metals. And it's an oxygen-based heavy metal chelator. And what you can see is that it totally prevents cadmium, copper, and zinc from being toxic, and it enhances the toxicity of mercury. So for those of you who do EDTA chelation, keep this in mind. This is a two-edged sword, the EDTA treatment of iron chelation in the body. And it's not just myself that says this. Chelation of mercury by EDTA enhances its ability to inhibit tubulin polymerization. And so here's the slide that the ADA hates. And what it is, we have 2 Alzheimer's disease subjects over here, and you can see that the tubulin in their brain is not photolabeling at all. Then we have 2 controls over here that photolabel tubulin quite effectively, and if you add less than a micromole of mercury, and this is all done very quickly, you can get with a 10-fold less than that if you let it sit overnight. It blocks it out, and it makes the normal brain look just like an AD brain with regard to nucleotide binding proteins. And note that it doesn't inhibit the binding of this particular protein. So what this strongly would indicate, and you can't deny it, that we know people get Alzheimer's disease. Everybody that does come down with AD, they cross that thin red line at some point in their life at different ages. You can't tell me that if you got 2 neurons left to rub together, that you can't look at this and say people who are going to get Alzheimer's disease, which is a huge percentage of our population, should not be exposed to mercury at all. It would definitely exacerbate that condition, especially mercury vapor, which crosses into the brain with a great deal of effectiveness. Now, here's the clincher, and the one that when people say, well, it's not that much mercury. Well, the amount of mercury that we found that causes this inhibition is a hell of a lot less than what you find in the brains of aged people. Mercury builds up in the brains of older people. And there was a publication done by the National Institutes of Dental Research and some people at NIH, and they measured the amount of mercury in the brains of nuns from Milwaukee, I think they were. And some of them had as high as 3 micromolar of mercury in their brain. A lot of them had over a micromolar, and that level is way, way more than you need to totally cause this type of disruption. I mean, the common sense isn't there. The chemistry is all on my side with regard to this claim. But let's look at it here. What happens with mercury? If we increase zinc, zinc is not very toxic. There's the zinc inhibition of tubulin polymerization. You have to go to very high concentrations to see any negative effect at all. If you add mercury in contrast by itself, it goes straight down. What happens if you combine mercury with the 10 or 20 micromolar zinc? And the difference is, if you start out here and you have this much zinc and this much mercury, and you increase the zinc to a higher level, you get this type of a curve. The presence of zinc tremendously enhances the toxicity of mercury. And that's a very well-known factor in mercury or heavy metal toxicity. You have synergistic effects. Cadmium, I use this one because cadmium was not nearly as clear. Cadmium just totally wiped out. You put a little bit of cadmium in here with this much mercury, and you're down here. And this fits the data published by Schubert a long time ago, where he showed that if you take rats and you give them enough mercury to kill 1 of 100, and you mix that then with enough lead that would kill 1 of 100, and 1/20th of that amount, you put those two together, you should kill 2 rats, maximum. And you didn't. You killed all of them. So that's what they mean by synergistic toxicity. The 2 metals together really dramatically increase the toxicity of the mercury exposure. So we can't, and the take-home lesson here for doctors and everyone is you cannot look at somebody and say how much mercury would make him toxic. It would take a lot less mercury to make a person with Wilson's disease toxic because of the excess copper in their body than it would a normal person. And so a person that's overexposed to lead, if they've been working around like I did as a kid on a farm, driving a tractor all day, burning leaded gas, and I breathe the fumes from it, you get a lot of mercury in your body. And that was proven by the research that preceded anything I did, that the lead concentration in Americans was very high, and it went down dramatically when we removed lead from gasoline. Because people like me, and I'm sure some of the others in this room here, spent a lot of time in garages working on cars, either for fun or because we had to make a living, and we were very high in lead. It was in our hair, it was in our bodies, and I think people who had lead and were exposed to mercury really got much more sick than those who didn't. And that's what the research, any way you look at it, does that. Okay, now, everyone, the major attack on this work from the ADA and others was that I was doing it on a brain that was in a test tube. So we decided to repeat this in rats. And this is a rather complex, not that complex, but it's a study that I did with the people at Calgary right before they did the neuronal work. But what we're showing here, this is a rat mercury vapor exposure on these experiments right here. We had control, a 2-day dose and a 14-day dose. And if you look at this, this is the level of tubulin photolabeling, and this is the level of mercury. Nothing happens. You got good photolabeling, no mercury in the brain, very low. And then we gave 2-day dose. The mercury level in the brain went up this high, but the photolabeling of the tubulin stayed about the same. I think this is an indication of how protective the glutathione in the brain can be. It does give you some protection. But if you went 14-day dose and the mercury level went from here up to here, the brain tubulin photolabeling went from here down to here. So if you pass a certain threshold, which can happen if you get overexposed, it goes around. And if you look at it, this is very similar to the Alzheimer's brain, whereas these two are very similar to the control brain in the ratio. And to make a long story, this is published. I don't think I have the reference on here. It's on the next slide. But what we showed was that the more mercury vapor a rat breathed after it crossed a certain threshold, The tubulin photolabeling went to hell in a handbasket. They had an AD-like brain. Well, some people said, well, maybe it's because it was causing the tubulin to disappear. But this is the immunostaining of the tubulin, which we do to quantify it, and you can see that the tubulin is there. This tubulin is very high, and its photolabeling is very high. The 14-day dose, the tubulin is still very high, but the photolabeling is controlled down. And in Alzheimer's disease, you get the same thing. The tubulin levels are not that different. There's a slight difference, but the photolabeling and the control is very effective in the Alzheimer's disease. It's dramatically reduced. So we could make a rat behave just like an AD brain. You know, you extracted the brains from them, and there's no end. Now, should this be a surprise? Look at this, the effects of sulfhydroreactive heavy metals on the in vitro polymerization of purified brain tubulin. This was done 1977, 1983, 1984, again 1984, before I did the work at the University of Kentucky. And this is on pure tubulin. And what they say down here, and this is what is really critical to look at, it says 1 millimolar EGTA, that's just another chelator like EDTA, prevented inhibition by copper and cadmium up to 700 micromolar, but did not prevent inhibition by either 25 micromolar mercury or 50 micromolar methyl mercury. So the chelators don't work with these guys in their research either. And down here they said addition of EDTA or EGTA, a 500-fold molar excess decreased concentration of mercury necessary to completely inhibit tubulin polymerization to only 2.5 micromolar. In other words, when you added the chelator, the metal became more toxic. Now, for those of you who do chelation therapy, EDTA in the blood, EDTA doesn't get into the brain very well, so it doesn't go in and affect this aspect. But your immune system has to have tubulin polymerization to be effective. And this is you've all seen this film, and I didn't want to show it because it takes quite a bit of time. This is the work done at Calgary. It's called the Calgary film. And what they did, this is tubulin, this is a neuron, and when they added 10 to the minus 9th molar mercury, this tubulin all precipitated or aggregated. You can see it just fold up, roll up, and get down here, and this kind of a abnormal looking thing here. And you see denuded neurofibrils, which lead to neurofibrillary tangles. So visually, you can see the formation of neurofibrillary tangles by the addition of nanomolar levels of mercury to neurons in culture. So now, so if we go back and we think about what I talked to you about, the structure of tubulin and axons, it explains, the hyperphosphorylation of tau, because other people have shown that mercury causes the hyperphosphorylation of tau, and it causes the formation of neurofibrillary tangles. It causes the destruction of the synapses. It causes the destruction of neural connections in the body. And so there's nothing remarkable about what I'm saying or what I have said. And I've been kicked in the teeth, many of you know, several times by the International Institutes of Health and Dental Research with regard to making these statements. It's outlandish. It's not outlandish. It's common sense biochemistry, and it's farm boy logic. It's just straightforward logic. Now, the only thing we didn't talk about was the beta amyloid. Beta amyloid plaques are the second most highly recognized diagnostic hallmark of Alzheimer's disease. And what we see here, this is published 2013 by Solomon Chu in the Toxicological Research, December 31st. And what they're looking at is here, this is beta amyloid level in these cell types, these PC12 cells, which are brain cells. And you see as you increase the concentration of mercury, that's 10 times 10 to the minus 9th molar, 100 nanomolar, and 1,000 nanomolar. You can see that the level of beta amyloid protein goes up, and it's actually very effectively increased at somewhere between 10 and 100 nanomolar. The mercury in your brain is a lot higher than that. Then there's not a person in this room that has just 10 nanomolar levels of mercury in their brain. Everybody would be between 10 and 100 or higher. And if you look at the plot of the beta amyloid level with time, and this is at different concentrations, you can see that it goes up very fast. Beta amyloid is produced by exposure to very, very low levels of mercury. Now, how does it do that? There's an enzyme. It's called NEP, or neprilysin. It's an amyloid degrading protease. Mercury and methylmercury both inhibit the expression of NEP, thereby increasing beta amyloid levels as seen in an AD brain. In other words, you can look at his NEP. The more mercury you have, the less of it you make. And this is the enzyme that breaks down beta amyloid so it doesn't form amyloid plaques. And if you look at the bar graph here, here's the relative density. Amyloid precursor protein went up. This is the protein that's used to make beta amyloid, but the production and the destruction of beta amyloid went down with increasing NEP. So, and if you added high levels of mercury, the NEP proteolysis of beta amyloid was decreased significantly. So this indicates how mercury causes an increase in beta amyloid protein. Now, if you know that the pharmaceutical industry has spent billions of dollars trying to find a drug to produce or remove beta amyloid protein from the body, from the brain, and they made antibodies that did this, and when they gave it to patients that had high levels of beta amyloid, they died. I mean, it was lethal. They had to stop the study. They had an antibody that would bind to beta amyloid and prevent it from forming amyloid plaques. And that seems to be part of the protective mechanisms. So the production of beta amyloid is a protective device, to protect us from low-level heavy metal toxicity exposure. Would be a reasonable explanation. But at this time, what we can honestly say, and you have to look at the NIH and you have to say, you guys you're either idiots or criminals. You have no choice. You can't be just misled. This is not hard research to follow. Mercury is a known neurotoxin. It produces every diagnostic hallmark of Alzheimer's disease at levels that are found in the brain. It produces all of the aberrant biochemistry, because we didn't show a lot of it, but creatine kinase is 95% inhibited in AD versus normal brain. And creatine kinase is an enzyme that we proved a long time ago is very sensitive to low-level exposure to mercury, sort of like this neprilysin. So you're having mercury get in the brain and produce the diagnostic hallmarks. It's easy to explain. It's straightforward biochemistry. You don't have to be a rocket scientist to find this. And why won't they, I say publicly, just if they want to invite me someplace to try and chew me up and make me look like a fool, just give me the invitation. I'll be there with bells on. Because I know biochemistry well enough to know I'm not wrong here. This is not rocket science. This is not, doesn't take any high level of mathematical analysis to get this far along. So we'll look at this, and this is on the slides, which all of you are welcome to, but we just talk about them. This is Oliveira did this work, and she did a test on 10 to the minus 9. That's 1 nanomolar mercury. Mercury increases tau phosphorylation and secretion of beta amyloid. She did this before the Koreans did their part. And this was published in Neurochemistry in 2000. We published ours back in 1988, so we were the first people to really show that. But what she did, kind of fit in with what we did. And this is long. This is neurofibrillary tangles. They show the neurons to 10 to the minus 7th to 10 to the minus 10th molar mercury rapidly causes stripping of tubulin from the neurofibers, forming neurofibrillary tangles, which is the diagnostic hallmark. And this is the one we just talked about. The 10 nanomolar of mercury inhibits neprilysin, the enzyme that digests and removes beta amyloid from the brain. This leads to the formation of amyloid plaques, a diagnostic hallmark of that. And therefore, mercury does, the through widely accepted, does a lot of things that when we look at it, we can say it would at least, if you're really a jarhead, you could say it doesn't cause mercury. It wouldn't cause mercury if you're exposed to it. But we know it makes you demented. We know the Mad Hatter from Alice in Wonderland was from breathing too much mercury, getting too much mercury exposure. It can cause dementia. And so when you look at this, you see what's going on. And so here we have a government that is sitting there looking you straight in the face. And I have talked to these SOBs straight on. And that's what they deserve to be called, because they're not good people. They're causing people to suffer dramatically, and this is something that we would really need to change. And if you read all the literature that's talking about how sick Americans are, we used to be a very hardy people, and now we're sick. Our children are sick. We have 1 out of 6 with neurodevelopmental disorders, and that's because we have a government just for the sake of producing and protecting an income property for the American Dental Association are allowing us to continue to put mercury in our children and in our older people, as well as inject it into their bodies with the vaccines. Mercury has no place in health treatments that I know of, except maybe in a thermometer. And here it is. This is something just for the people who are new here. The amalgam fillings are 50% mercury. And mercury, is it's a liquid. And you mix it with metals, it forms a paste that turns into a very hard material. And these are called metallic bonds. Mercury forms a weak metallic bond with other mercury atoms. But when mercury binds to tin, it forms a tougher bond, because it's just chemistry. I mean, I can't explain that to you since you guys haven't got the background to understand it, but you can understand. They're called metallic bonds. But the bonds that mercury makes are weak, and so it's slowly evaporating off of your amalgam filling, and if you put pressure on it, you put heat on it, it'll come off faster. That's what any chemist that knows anything about metal chemistry would explain to you. And that's what happens in your amalgams. The more you chew on them, the more hot coffee you drink, the more it comes off, the more other metals you have in your mouth, the more galvanism you have in your mouth, the more mercury that's released. So there's nothing safe about this, and they can't make any type of argument that holds water. And I've had amalgam fillings described to me that the mercury in an amalgam filling is like an egg in a cake. It's not a cake. It's not an egg anymore. That's stupid. Mercury is an element. You can't break it down. Yes.
Well, I have to say, in January of 2009, the Norwegian government banned mercury out of Norway, and the health minister said specifically, including dental mercury. The Swedes, not to be undone, did it 3 months later. So civilized countries have recognized what you say and have acted.
Yeah, I was on the Swedish heavy metals biology board for a long time there, and that's the reason why my company is being developed in Sweden, because there's a level of intelligence and honesty there that we don't have in this country with regard to health administration. Thanks. And this is the clincher, and we've done this. We've done this in front of Congress, where you take an amalgam filling and watch the smoke come off. David Kennedy has a nice film on this on the IABDM website, and you can see the mercury coming off, and they try to tell me it's water, and you're doing it with UV. Hey folks, I did UV spectrophotometry all my life, all my research. Water doesn't absorb UV waves. If it did, you couldn't measure the spectra of a nucleotide in water. So this is a 254 nanometer wavelength light that's being absorbed. And you can see this, you can show it, and you can put a mercury vapor sniffer right in the center of that. And, pardon me. And show that it comes off thing. And I've demonstrated this for television several times. And they accuse me of faking it. Well, I don't have to fake it. I mean, hell, I'm not the only person that does it. If I was the only one that could do it, then you have to say, well, he's got fast hands. But I'm not the only one that can do it. So anyway, this is, I guess what I'm trying to embarrass our government. I'm hoping there's somebody from the FDA sitting there realizing what a bunch of dumb corkers they look like. I mean, and you wonder why parents in San Francisco, they say, are not vaccinating their children. From the rich, educated districts, not from the poor people. The poor people are still vaccinating. But the rich people are refusing to vaccinate their children, mainly because they can read, and they have computers, and they get on a website, and they're finding out that our government is less than honest and candid about the toxicity of mercury. This is, and I want to point out, notice the scale here. The scale goes up, it doesn't go up linearly. This was done, mercury levels by anatomic site and number of occlusal dental amalgams. And the dental amalgam and mercury levels in autopsy tissues. And what it's saying is that if you look at this and realize that this is a lot higher than it looks on the scale here, and so is this, that when you have more dental amalgams, you have more mercury in your tissue, and if you think that's good, then you shouldn't be in this room. And it tells them, when they say, well, the mercury doesn't get out of the amalgams, doesn't get in your body, you know, you're just ignoring all the science. And when you look at the science and the number of people that have done this, you realize that the dental branch of the Food and Drug Administration are a bunch of liars. You can't call them anything but that. I mean, why do you get educated if you refute the value of your education? And this is something that goes on, and this is when people who want to control science don't know any science. This is the other experiment, because they were trying to tell me that, well, amalgams tied the mercury up and didn't make it toxic. So I had a dental friend from Colorado send me a tooth with a big amalgam filling in it. I washed it extensively. And then I took that tooth and I put it into a mill of water or something like that. I forget if it was a mill or 2 mills. And just let it sit there. And then every so often I would take out an aliquot of that water and see if it was toxic to brain proteins. And this is the result. The result is, this is before I put, this is in the distilled water. And this is in the percent of GTP bound to tubulin. And you can see within 1 hour, that water was so toxic that it inhibited over close to 80% of the tubulin in the brain. And that was a tooth that came out of a human's mouth. And it was old. It wasn't a brand new one. If you put in a brand new one, it would have even been worse. So whatever comes off of an amalgam filling, whatever the ADA wants to tell you it is, you have to understand it's toxic. It's incredibly toxic. Anybody can do this study. This is the essence of this study. This is looking at creatine kinase by exposed to water exposed to the same aged amalgam. And you can see that this enzyme is inhibited 95% in Alzheimer's disease brain versus age-matched control. And it makes creatine phosphate, which is used to make ATP and GTP and everything else. And you can see that if I leave it in with the control, the enzyme's very active. If I put it in 15 minutes with the amalgam, 1 hour, that I get total inhibition of this enzyme. So here's another key enzyme, creatine kinase. It's brain creatine kinase is essential for good neural transmission and good brain function. And amalgams inhibit it exactly as you would expect if they're releasing mercury, because this is a very mercury-sensitive enzyme. So what we found here, and this is, matter of fact, was published in Molecular Brain Research in 1998. Over 95% of the NAD brain and rapidly inhibited by mercury, dental amalgam mercury. It has a very reactive thiol, because I was a guy that pulled out the active site peptide on this, and we did this in biochemistry in 1994. So in addition to the production of the three diagnostic hallmarks of Alzheimer's disease, mercury also creates the unique inhibitory abnormal biochemistry you see in the NAD brain. And why this escapes our government, because I've been yelling at this, some of you know, for 20 years. So I think the problem isn't a lack of science. The problem is the lack of a government that is really honest. Our American health agencies are not at all honest. They're controlled by moneyed interests that have a vested interest in not being sued, or to be allowed to continue making a cheap product that they can sell, and not being have to face up with what they've already done.